Hello and welcome to Inside Exercise. Today I've got the legend, the icon, Claude Bouchard, to talk about genetics and exercise. He's the one that everyone thinks of first, of course. He's been doing this for 40 or 50 years, he said. He's recently retired at 82, still going strong, as you'll see. From Quebec, you can still hear a bit of French accent, but he's been in Louisiana for many, many years. The major focus was the variability of response to training. So some people increase their VO2 max and performance a lot when they train. Some people don't. Some people have a high VO2 max basally. Some people don't. So how much is the genetic contribution to that? We also discussed health issues. So for example, insulin sensitivity, how much of that variation is due to genetic differences. So a lot of discussion about his classic heritage study, how he started off looking at animals before that. The sort of classic studies about responders and non-responders, high capacity runners, low capacity runners, etc. So I think you'll really enjoy this one. Hello, Claude. How are you?
Good morning or afternoon. I'm good, thank you.
It's morning for me and afternoon for you, I think. You're looking well. I was a bit worried. You told me you had COVID and then you were going to go fishing. And I thought that's not bad for someone who's 82. Normally someone gets COVID, you've got to worry a bit. He's like, oh, I can't do it now. I've got COVID, then I'm going fishing. So how are you?
I'm good. I am 83 though. I got 83 recently. COVID was not too severe. It was really mild because I'm fully vaccinated and I have had the boosters. A couple of days of sneezing. That was essentially this. But the fishing was a challenge.
Fishing was a challenge or wasn't?
It was a challenge. Very big, demanding river. Lots of rapids and very tough river to fish for a fly fisher.
Yes, so tell me about this. So when we spoke beforehand, I said, what do you do now that you've retired? And you said fly fishing. And I said, oh, so you go fishing. And you said, not fishing, fly fishing. So why don't you tell them, this is demanding, is it?
Well, it's different. We like to think that it takes skills to become a good fisherman. You got to work at it. And I've done it for about 60 years now, mainly for trout and for salmon.
Okay, so I guess you're good. So you're flicking the line over the water. You're not just like letting it sink to the bottom and like sit around and chat.
Among other things.
Oh, okay. All right. Well, maybe we need a whole new, you need to go on a fishing podcast, maybe. All right. So actually, where actually are you? You're in Quebec. You've been in Quebec, but you're in Louisiana nowadays. Is that right? You have a French accent.
Yes, I am a native of Quebec City in Canada, the French-speaking part of Canada. And but I have been working at the Pennington Biomedical Research Center in Louisiana for the last 23 years. And I retired from that position about 8 months ago.
Yes, so 82, you thought that's time to retire. So you've had an amazing career. I'm just wondering, how did you actually start way back then? Were you always interested in genetics? Were you interested in exercise? Like, how did you actually get into this area?
So it is a long story, but I will make it short for you. I became fascinated early on, before I did my doctoral work. I became fascinated by the observation that there were large human variability in cardiorespiratory fitness among sedentary people. Confirmed sedentary people, we had a sometimes threefold range in a group between the low level of cardiorespiratory fitness and high, and they were all sedentary. So that level of human variability was intriguing because they were all young males. We're not talking about having social demographic characteristics. I will explain this. And they were not obese and lean, or they were, recreationally capable of being active, but they were not. And that was, it took me a while to decide to focus on genetics. It was not on my radar screen. It took a while. And once I decided that this would be my area of work, then I went on and did my PhD at the University of Texas at Austin, so that I could develop a good base in genetics from the molecular, the biochemical, the immunogenetics, and then the human genetics and the population genetics. I covered the whole waterfront to be able to develop a comprehensive research program. Took me years to get there.
Okay, and we have to remember that this is all before, nowadays we know about, a lot more about DNA. We can measure it. It's actually cheap to do a whole genome. Back then, how were you piecing together the genetics? So obviously you had this variability, but how did you start trying to piece it together? How much was genetic? How much was?
So we're talking here about the mid-70s. That's when I began to focus on genetics and launched my research program in 1977 in this area. And at that time, we were not able to genotype people based on their DNA. You're right, we were not. So we were using two different strategies. One was to look for naturally occurring informative situation, like biological relatives, but related to variable degree of relatedness, so that we could begin to infer, based on the magnitude of the variance partitioning, could infer about the role of potential genetic transmission, maternal effect, paternal effect, etc. That was the first line. We complemented this with a number of so-called genetic markers that were commonly used in medical settings, like the blood groups, the HLA system, the histocompatibility system, tissue compatibility. And we developed electrophoretic procedures to separate proteins from the blood and the muscle based on their charge and on their mass, so that we could identify whether there were variants among people, not at the product level. So over a period of about 6-7 years, I ended up having more than 60 genetic markers in my lab at that time. And we were so pleased with ourselves. We had 60 genetic markers. We thought that
60 or 16?
60. A little bit more than that.
It still sounds like a lot to me, actually.
Yeah, but this looks so naive by today's standard, but that was progress. We were moving, we were able to track DNA variants from one generation to the next, and we were trying to see whether this correlate with what we were seeing in the transmission of the traits of fitness or body composition and other phenotypes. So that was, a long way from where we are today.
I'm just trying to understand that. So what were some of the variants there? Because I'm just thinking about, measuring, I'm sort of assuming measuring DNA, but you weren't able to do that. So what were some of the things you were measuring in the blood, did you say, in the muscle? What were you actually measuring back then?
So the blood groups, for example, starting with the ABO blood group, the RH system, the Duffy blood group, the MN blood group, there are about a dozen of them that we were measuring in people. And then for the histocompatibility system, we had 4 different loci that we were measuring. And then we added the proteins from electrophoresis.
Okay, so what would you actually be doing with that? So you'd have these people you'd bring in, you'd do their VO2 maxes, you'd have, paternal twins, fraternal twins, cousins, whatever. And how would you then relate that to those 60 measures? What would you be doing then?
Well, we were looking for we were genotyping, so we had a full operation of genotyping people from a blood sample. And we were using the genotype. For example, if we take a blood group where we have only one allele, so that will give us two alleles, a major and a minor allele. Give us three genotypes. So we would look at the differences between the three genotypes for, let's say, maximal oxygen uptake. Now, that's a long shot because we know this is not likely to be a gene that contributes directly to maximal oxygen uptake. But the rationale was it might be inherited together on the same chromosome with other alleles that are relevant to maximal oxygen uptake. Okay. So having about 60 genomic markers was a step in the right direction. Even though we did not know what were the important genes, we were trying to identify them by defining positions in the genome.
Yep. Okay. So obviously now you have, thousands and thousands of genes that have been able to, through linkage studies and whatever. So do you think, how close are we now to sort of, because I still assume it's a multi-gene, because, for VO2 max, you're going to have your cardiac output, which is very important, obviously, but your oxygen extraction, your various other things that play a role. How close are we to actually look at it and go, oh, you've got this gene that therefore your VO2 max will be this. We're not there yet, I assume.
Well, of course, the physiology has improved as well. We know a lot more about the physiology of these traits. So we have guiding, we have a guiding biology to assist us in our search for the genes. Now, the genomic pursuit is now a totally different ballgame. We have not hundreds, not thousands, but millions of genomic markers that we can genotype in every person. And we can also sequence them completely. Whole 3 billion bases of DNA, we can sequence them. Right. So we don't have a marker, we have the whole book for each person in a given study. So
that's from too little to too many now. It's,
well, in a sense, there's some truth to that. It's enormous. Now, some of that variability is not of importance to any fitness trait. But I'm sure that in there are lots of very important variants. And the science of the last 20 years tells us that these complex traits are influenced by variants in our genome that are extremely numerous. We're not talking about 5, 10, 50. We're talking about hundreds and thousands. So defining this profile for a given individual is not something that you do on the corner of a table. You need a sophisticated lab and genotype, and then you need the proper computational biology and bioinformatics to identify these important kernels of truth in this mass of data. The research is going slowly on this, but it is moving absolutely in the right direction. We see that the cohorts are getting better. We have larger cohorts, more powerful genomic studies, and the field is progressively bringing in expertise that we don't have in the exercise science, but are absolutely essential if you get involved into this kind of work. We need people who have a good understanding of the bioinformatics and the computational biology. Otherwise, you don't know what you're doing, really.
Yeah. Okay, so when I think of you, I always think of the heritage study, and I'm just wondering, how did you actually lead up to the heritage study, and why don't you actually even explain the name heritage, how you got that name as well?
Well, we you know, when I started my genetic endeavors, we did not have enough background data to justify a full large-scale project with very expensive funding. So I launched a series of studies with the support of monozygotic twins. 4 different studies, actually, with pairs of monozygotic twins. The rationale was that we cannot study the genes directly yet in people, but we can try to replicate some of the things we do in animal research, where a lot of the foundation of human genetics has come from. And in this particular case, it would be the comparison of inbred strains, a strain where in each strain, all the animals have the same genome. And differences between strains at that time were thought to be largely explained by genetic differences between the strains.
Yeah.
In people, it would be nice to do that if we had large
Yes.
sibships, you know, with 5, 10 identical twins, but we don't have that. We have pairs, and but we can have many pairs. That's an advantage too. So I did a lot of those studies to try to see whether there was enough evidence to justify a bigger project. And quite clearly, all these studies, one was on high intensity intermittent training, one was on endurance training, strength training, etc. We did a series of these studies, and also exposure to overfeeding, or the opposite, negative energy balance, and cold exposure as well, so that we understood that the human physiology was clear. There were differences in the ability to adapt that appeared to be caused by genetic differences. And armed with this data, then we went to the National Institutes of Health of the US, and I was still in Quebec at that time. And we I recruited a few of my colleagues in the US to work with me on the heritage study. And 5 of us made a large application to NIH, and we were successful, fortunately. And the NIH funded heritage for, 20 more years.
So tell us what heritage stands for. It's a very clever name, because it's, you know, it's the meaning of the word heritage, but also how does it break down the name heritage?
So it says health, risk factors, exercise training, and genetics.
Yeah, yes, that's a clever name, right? Because it makes sense when you break it down. It makes sense with heritage in terms of genetics.
We liked it too.
Yes, and it's good to have a catchy name for the NIH, I'm sure, as well. So can you give us a bit of an overall feeling of what you actually found in those early studies, what percentage you felt that genetics was playing to your initial VO2 max, yeah? And then what the data is actually showing now when you've got billions of genes and whatever. Is it holding up, and what sort of percentage are we looking at?
Okay, well, a few highlights, because you know, there are hundreds and hundreds of things that we have found and reported. But I think that from all of these efforts in twins and heritage, the family studies as well, one major finding is that for cardiorespiratory fitness, there is no relation between your level of fitness in the sedentary state and the way you respond to an exercise program, to a fixed dose of exercise. Zero correlation, which is extremely important biologically, because it says that the biology of one is different from the biology of the others, completely different. And the findings that we have thus far are supporting this completely. That's something that we have found in the twins, and that we documented at a bigger level in heritage. And it was true in men, women, young and older, and blacks and whites. There is no relation between the fitness level that you are exhibiting in the sedentary state and how much you respond when you're exposed to a standardized dose of exercise.
Wow. Okay. I have to say, I had that totally wrong. So I thought that there's something like, 25 to 50% of your initial VO2 max is based on your genes.
So,
And then how much you adapt to training was also based on your genes. I thought that was from the high capacity runners, low capacity runners stuff. I thought that
they are both influenced by your genotypes, but not the same gene, not the same genetic profile. They are different, a totally different biology. And we can expand on that later if you want, but that's something that we saw clearly. The second important observation is that, and that speaks more to the genetics, both in the twin studies and in the heritage, we saw evidence for familial aggregation, concentration of the status in the sedentary state or the response level in response to the exercise training program. We saw a concentration of high responders or low responders in some families and the opposite in other families. It tracks along family lines. And that is supportive evidence for a role of the genes. It doesn't say this is caused by gene X, Y, or Z, but it says the underlying cause of that is quite likely to be found in your genome. And that was unanimously supported across all the twin studies, and of course, better documented even in heritage. And this familial aggregation accounts for about 50% of the variance.
Yes.
In maximal oxygen uptake in the sedentary state, and also about 50% of the variance in the delta, the response to the exercise program to which we exposed the heritage participants. So that is those two observations are extremely important because they are essential to organize the search for the genes. You don't put together sedentary people and active people in a sample where you search for the genes. You're going to find a set of confounding signals.
Mm.
You got to work with people in the sedentary state and a delta study where you have the true response to the training.
Okay. All right. So, and that is holding up. So what your initial studies with the, when you didn't have all the amazing methods that you have now, and the studies you have now, they are fitting, that you're getting about the same sort of numbers?
Well, we're not focusing on numbers anymore because we're satisfied that there's a genetic component to sedentary state and trainability.
Yep.
And it doesn't matter really in the end whether it's 45 rather than 50 or whatever. It doesn't really matter.
It's the same ballpark.
Yeah. What we are interested in now are the genes, the sequence differences, the impact it has on the gene products and the pathways and how these genes interact with one another, and there are big lessons there already. Big lessons. One is that in the sedentary state, what is emerging from these complicated analysis of the genome, plus the proteome also, because we do the protein as well in the blood, and the metabolome, and the transcriptome from the skeletal muscle. What is emerging is that we're recapitulating what the physiology has told us. The main component is the stroke volume, cardiac output, the blood volume and the hemoglobin content, and then the skeletal muscle machinery with the myoglobin. The pathways that are coming out most strikingly all relate to this biology, transporting the oxygens to the muscle for work. But the dominant is the heart and the stroke volume.
Yeah, yeah.
In contrast, for the trainability, the dominant pathways relate to the ability of the organism to grow, to expand.
Okay.
We have variants in genes that are typically active during embryogenesis, during growth of tissues that are emerging. That is as if they were preparing the organism to meet the demand of a new challenge. That's what the exercise training program is. It imposes a demand that cannot be met just by having the typical maintenance gene that were needed in the sedentary state. They are not sufficient when we train. So yes, in the end, these genes will augment the stroke volume, the cardiac output, the blood volume, the hemoglobin content. They will ultimately end up there. But the machine is getting ready to grow to meet a stress of exercise training. So that is really fascinating.
Sure.
And they are not correlated. For example, at the protein level, we have in the blood about 150 proteins that are related to maximal oxygen uptake in the sedentary state. Out of 5,000 proteins we assayed in the blood, about that number, 150, are related to differences in maximal oxygen uptake. For the delta, same trend, we have about 100 proteins there related to individual differences, but only 5 of them are common between the two. Different biology.
Okay, okay.
So these are big lessons, and I think we cannot anymore justify conducting studies of fundamental biology by combining people who are sedentary with others who are moderately active or recreationally active. It has to be clean. Sedentarism is very different from the physically active mode of life.
Yeah, yeah. Now, how much of, we've been thinking about genetics along up till now, obviously, but I guess once you start exercise training and you're active or inactive and things, you're starting to affect your epigenetic background as well, right? How does that, I guess that's another layer of complexity.
It is, and I think the jury is still out on how critical that is. It probably will turn out to be quite important, but at this time, I don't think we have the data. We have some papers on the epigenetic changes in the blood. Well, that's something, but the action is generally not in the blood. It's more in the heart and in the muscle. And we would like to have epigenetics done there, but that's not always, certainly for the heart, it would be a challenge, except it could be done in models. But in humans, thus far, I think the jury is still out. There are suggestions here and there, and don't forget, like in any field of research, the first couple of papers that find something positive, they get a lot of attention, as if the effect size was large, big, but over time, the pendulum tends to swing back in the middle, and we don't know where we are now with respect to epigenetics. But I'm pretty optimistic that we will have data that would justify incorporated epigenetics in the model, in the training response model. Whether they will play a big role or a moderate role or a small role, I think remains to be determined.
You've had, I was thinking, you've had to be very adaptable yourself. If you think about over these last 50 years or whatever, how things have changed, yeah? So just, so when you said about, you know, some studies have shown this and some studies have shown that, I'm wondering what things have sort of, you know, been a struggle or haven't worked out the way you thought, or, you know, whatever. I'm assuming you've done a lot of troubleshooting in your day.
You know, the first big set of hurdles that we had to face was related to technologies. Obviously,
yeah.
My lab went through, I would say, 4 or 5 generations of different technologies. Started with, like I said, the blood group and the histocompatibility, and then move on to electrophoresis, and then restriction, fragment, length polymorphism of the genome, and then microsatellites, and then.
And it's probably getting more and more expensive as well, right?
And then now it actually is becoming cheaper.
Oh, it's becoming cheaper now, but what I mean is, as you had to do all these more methods, I'm sure they were expensive before they got cheaper.
Yeah, you're right. They did, but now they are much cheaper. Yes. But you know, these technologies require a different analytical tool as well. You cannot analyze the data that we had with the blood groups and others, the same thing as we analyze the microsatellite data with the linkage approaches. It's totally different. And now it's, again, new technologies all the time. So very few labs have been able to adapt, I think, throughout this journey with the same human resources. We have had to incorporate
different expertise
all the time.
They're probably saying, hang on, Claude wants another machine. He wants different, you know, skills. I mean, it must have been crazy.
Yeah, well, it was, I had a fairly large lab for quite a while in Quebec. So it was manageable to add 1 person or 2 because I had about 60 people in the lab. So adding 1 or 2, or the turnaround of the people in the lab was okay. Well, yeah, with technicians and postdocs, 6-0, yeah.
Holy mackerel. Okay.
Yeah, that was a productive period. So it required that we constantly had the people coming in with, you know,
yes,
who knew how to do X and knew how to do Y, and then they would enrich all the projects.
Yep.
All the discussion.
Okay, so earlier I mentioned the high capacity runners and low capacity runners. Do you want to just explain, you know, these are rodent studies. Do you want to just explain a little bit, you know, what they are to people that don't know about that, and how that's sort of fitted in with what you found with your human studies?
Okay, so those were not my studies. They were mainly those of Steve Britton and Lauren Corr, who worked together in Ohio and then moved to Michigan, and the University of Michigan. And it was very astute. They bred for high capacity running and low capacity running. They bred rats, and in an outbred strain of rats, where you had genetic differences at the onset, just as we have in people. So they bred at the upper end of the curve, males and females who were the highest performer in the sedentary state, and the same at the low end.
That's the important thing that people don't always get. So these have never done any exercise training. They just live in their cage, and they just get the ones that are naturally good runners, and they breed those with the ones that are naturally good runners. And they're now up to like 25 generations or something, aren't they?
Oh, they went to, I think, about 30 some generations.
Yeah.
So over time, the low capacity runners bred only low capacity offspring. Mm-hmm. There was no change over the generation in their performance time. It was an endurance performance. But the high capacity runners over time concentrated the genes of high capacity running among the members of the upper end of that distribution. And the performance of the high capacity runners increased by about 400-fold, 400%. So that is an enormous difference. Yeah. So it seems that the low capacity running genes were pretty abundant because it didn't matter. Breeding did not change that. They remained low. They didn't go much lower. They remained flat. But they were distributed in the genome of these rats. Some genes had an impact on high capacity running, and they were concentrated over time. Genetically, the two strains diverge more and more and more and more from one another genetically at the gene level over time. So that was pretty remarkable. And then more recently, they did the same thing with training.
Yes.
So they started with this strain and had a low response and a high response to a training regimen. And they bred for training response, found the same thing as they did. But we don't know in the rats whether these two are correlated or not. Like in people, they are total zero correlation, but in the rats, I don't know. I don't think they did that study.
Okay.
So it was going on at the same time as heritage was going on, and we had discussions with Steve and Lauren regularly, and yeah, our results are entirely compatible.
They are.
So their rat model was really a good model because it really mimicked what we see in people.
Yep. So if you, again, whether it's a rat or a human, because I think they're both showing the same thing, if you have someone who is a high capacity runner, so you get them in the lab, they've never done anything, and your VO2 max test and they come up at 55 or something, I've seen this myself, and I go, hang on a minute, you sure you don't do anything?
Yeah.
Do you think that person, do you think they are going to, are they the person that's likely to do very well? Like if they've got that low base, we haven't talked about responders and non-responders yet, but I'd like to think on average, most people respond. So do you think that that person that ends up being a very good endurance athlete, for example, do you think that they are sort of like a high capacity runner to start with, and then you train them? Or do you think you can get sort of anyone and train them on? My feeling is you've got to have a bit of natural grunt already. Is that a fair point? What do you think?
Well, they are, in the case you described, they start with a high base. That's very encouraging. But there is zero correlation with the training response. So they could be flat. Right. And we see that in heritage. Absolutely. So the ideal situation, if you're interested in performance, is to be both. Have a very high base in the sedentary state and be a very high responder. That's the ideal. But the correlation
is zero.
The group biology is against you.
Okay, so across a population, that would be the case. But do you think if you went to like Eliud Kipchoge, the guy who ran the sub 2 hour marathon, in Vienna, you think if he'd never done anything, surely he would have a high VO2 max? Yeah.
I have no idea.
No idea. But okay, I know you can see where I'm heading here. My feeling is, I'm not a genetics guy. My feeling is that surely these world-class endurance athletes, I know there's no correlation, but they must be the lucky ones that have come along and they've got a high natural VO2 max and they've had a high. Is that fair? Or are you saying we don't really know that answer?
Well, when you talk about people who have a maximal oxygen uptake of 80, 85, 90, gosh, there's no way that they can reach that without starting from the high base and being highly trainable, for sure.
Yeah.
I don't know about those who are in the 65, 68, 75, 70, 75, 78 range. Okay, there may be. They may not be in the top 1 90th percentile of maximal oxygen uptake in the sedentary state, but they certainly were not in the lowest decile.
Yeah.
But it's hard to tell, except maybe in the very high ones. There's no way you can reach that.
That makes a lot of sense to me. And you know what I like about that while you were saying that? I like the way people can't just say, oh, look, I can't run a marathon. I don't have the genes, you know, because it means that everyone should be, I mean, sure, you might have terrible injuries or whatever, but it means everyone should be able to get a decent VO2 max. You know, they may not get it 85, but everyone should be able to train. You know, what I like about it is you can't just, you know, if you put in the work, you'll get the results on average.
But that's not exactly what the science says, though.
Non-responders. Yep.
If you train for 20 weeks and you have zero improvement, and you have members of the same group who did the same training as you did, and their VO2 max improved by 50, 60, 80%, you'll have a hard time raising your VO2 max to a high level. If you are non-responsive to one dose of exercise, you might be able to get off the ground and get some training effect, changing the exercise prescription. Yes, I agree. Although this remains to be shown, but I think we can probably move the needle, but we're not going to move the needle from being a non-responder to being a super responder.
Yes.
No way.
All right. So this is where we've been heading towards this. Anyway, I still don't want people to have the excuse. Can we just agree that people shouldn't be able to say, oh, I've got crappy genes, there's no use me exercising? Can we agree on that at least?
Oh, we certainly, I am in full agreement with this for several reasons, but the main one is that there is zero correlation between your gain in VO2 max and your improvement in insulin sensitivity, in HDL cholesterol, in blood triglycerides, and inflammatory markers. It's the change in fitness do not predict your improvement in your health profile. It's the exercise that makes the difference for the health profile.
Beautiful. Yeah. Okay, beautiful. Yeah, I was wanting to head towards that because we've been sort of focusing, I've been focusing on VO2 max, and I was going to want to get to the sort of health side of things a bit later on, but that's interesting. So you're saying your ability to increase your VO2 max is actually also not correlated to your ability to improve your insulin sensitivity and things like that. Is that what you're saying? These are totally different as well.
Absolutely. We just actually, we've known that for a while in our studies, but we just published a fairly comprehensive paper on that in the British Journal of Sports Medicine. Okay,
so this fits really well with because, you know, we often talk about metabolic fitness and aerobic fitness and whatever, and these are sort of different things. Okay, so just to get back again to the responders and non-responders thing, you know, again, it's shown, I think, with VO2 max and with health things. So I think it's Cherie Kohlberg showed something like 15 to 20% of people with type 2 diabetes, she found were sort of non-responders in terms of their insulin sensitivity, I think. Is that right? And then you've also got the non-responders in terms of aerobic capacity. Yeah. To me, I'm always surprised by that because I always feel, but it sounds like the more we talk, I'm probably wrong. I always feel like surely if you train someone hard enough, or you tried a different type of training, so you did HIIT, or you did strength training, or you did whatever, surely they would respond in some way. Because personally, I never see non-responders, you know what I mean? But I haven't done huge studies. Can we kind of tease that out a little bit? Are you saying there's people that don't respond to any sort of training or just to some types of training?
Well, we don't know that. We don't know that. We suspect that we can move the needle if we change the exercise prescription. That's as I just said before. We could probably do that, but we don't have data. But what we know, though, is that even though there may not be a gain in maximal oxygen that will be, you know, substantial, like a 10-15% increase in maximal oxygen uptake beyond the error measurement maximal oxygen uptake. there are beneficial changes in cardiorespiratory fitness, but not the maximal value.
Yes.
You have an improvement in some maximal exercise capacity.
Yep.
Your blood pressure, your heart rate, your pulmonary ventilation, etc., gets better, easier at some maximal levels of exercise.
Yep.
But your maximal oxygen uptake does not improve. Your maximal power output will go up a little bit because you're mechanically more efficient. You've trained for, 6 months. You are better coordinated. Your power output will go up, but the maximal oxygen uptake stays flat. But that should not be a total discouragement because your fitness has improved, not just your maximal oxygen uptake.
Yeah.
Your fitness in general has improved, and your health, your cardiovascular and diabetogenic risk profile has improved also, quite likely. So everything is not lost.
Yeah, and it just again shows the complexity of everything. Yeah, so you can have someone that's, if you say, that person's a non-responder, you've actually got to sort of define what you mean. Like, they were a non-responder in terms of their VO2 max, but as you would expect, at submaximal levels, their lactates were lower, or they had a lower RER or whatever. So it doesn't. If someone's a non-responder in some way, as you said, they might respond metabolically with sensitivity or something. So again, we're back to do the exercise. There's no, you just do it because it's good for you no matter what. Yeah,
absolutely. It's, we have applied, I don't know why, but in the literature, people have had the tendency to apply this concept of responders and non-responders to a performance, maximal oxygen uptake. But in reality, there's a lot more to exercise biology than maximal oxygen uptake. And we can be a poor responder. I'm not calling them non-responders totally. They're poor responders because if we were able to train them differently, we probably will get some improvement. We will not transform that person into a high responder, but we'll get some improvement. But if you look under the hood and you look at the cardiopulmonary response and the hormonal profile, the substrate utilization, you will see a lot of benefits.
Hey, I had someone called Catherine Smits on here a few weeks ago. We were talking about exercise and cancer. And she somehow you came up and she said, a good question to ask Claude Bouchard would be, with exercise and responses that people have with cancer, whether you're preventing it or improving it during the cancer or reducing the chances of getting cancer again, there's, the variability. So I wonder if anyone's thought to look at that sort of concept of,
Oh boy. I will invoke the fifth. I am not an expert on that. I published with Alejandro Lucia from Madrid several years ago, a paper where we were trying to put our mind together around the importance of cardiorespiratory fitness for cancer prevention and cancer survival. And that has been, I think, my only cancer paper in my career. And based on the data that we were able to put together, it appears that the higher the VO2 max, or the higher your exercise intensity when you exercise, the better the protection seems to be against cancer or cancer recidivism. But the data are really limited because the population studies that are required for these kinds of observations do not generally measure maximal oxygen uptake. And they are not exposed to high intensity training program. So it's suggestive, but because one of the reasons we were finding this kind of indirect evidence intriguing is that when you exercise, of course, you secrete a lot of cytokines, myokines for the muscle, adipokines, cardiokines, hepatokines. The more intense the exercise, probably the more abundant are these cytokines in the circulation. And some of them growth promoting cytokines, for sure, because you are imposing a demand, so you're imposing growth. That's scary a little bit for a tumor.
That for cancer, yeah.
But others are there to check on growth also. So it's a push and pull thing. So yeah, it's an area worth exploring, but it requires some pretty sophisticated biology and a lot of money.
I wonder if one day, you know, you could, it would just be incredible to get this point, but you could have someone who's got cancer and say, oh, they're likely to be a responder to exercise. So let's really, you know, try and encourage them to exercise, you know, to prevent cancer, or, you know, to prevent it coming back. And then, oh, this person is probably less likely to respond to exercise, but they might respond to nutritional, you know, that would be, you know, pretty amazing, but it would be extremely complicated.
Yeah, but I don't think there's anything to lose to recommending exercise. In all the studies I have seen about exercise and cancer, sometimes we see no effect, but I've never seen a study where it was a negative effect. It was a detrimental. So I think to put all the chances on your side, you know, the most prudent position is to exercise, and for secondary prevention at least.
Absolutely. So just thinking about, it's interesting. I've learned a lot already. So I didn't realize that there's no correlation between your initial and your adaptations, etc. That's really interesting. Just trying to, as again, we've talked a lot about VO2max. Is that the same with your health? So if you look at your starting health and then your ability to improve your health, so insulin sensitivity, etc. Is it the same sort of thing in terms of the genetics, do you think? That there's no correlation between your initial and your ability to improve it with exercise?
It depends on the health traits. No, it's not true. For example, HDL cholesterol. If you have high HDL cholesterol, and to begin with, and you exercise, you're not improving more. You tend to stay about where you are, while those who are low tend to improve. Blood pressure, those who have high blood pressure, elevated blood pressure, not clinical definition of high blood pressure, but tend to trend high. They exercise, they reduce their blood pressure more than those who are low. So there's a correlation for some traits, none for VO2max, but for some traits, yes, there is a correlation. They're not all the same. The biology varies from trait to trait.
Okay.
That's what Heritage has told us. Yeah.
So okay, so you've retired, yeah, which, you know, at 82, I think that's a reasonable. 83 now, but you are retired at 82. I think that's a good time to think about slowing down. So if you were 50 now, and you were like thinking, okay, so where should I be heading? You know, so where is the research heading, do you think? What would be, you know, we touched on epigenetics. What do you think? Where would you be most excited about, you know, what needs to be looked at in the next years?
Well, I would certainly, if I had about 20 years of career in front of me, I would not hesitate in incorporating the epigenetics together with the genetics, because they are interconnected. The DNA sequence variance that is the basis for a genetic influence in the population is also quite likely related to the epigenetics, because these variants in the genome, the sites that are known to be the subject of epigenetic changes. CPG island in particular, for example, DNA variants introduced in these islands change the sequence. So you create or you destroy epigenetic sites with the sequence differences. So the two of them probably will greatly benefit from being looked at together all the time. And in exercise studies, I think this is something that has not been done on any scale yet. So I probably would contemplate approaching this, but I would need to be in a team where we have extremely good bioinformatics and computational biology support.
Yeah, okay. So is there, I'm just trying to think, is there anything we haven't covered? I feel like we've sort of covered a lot of stuff. Is there anything you'd like to sort of bring out that we haven't covered, you think?
I think we have a biggest message are there. Yeah, we covered the most important messages.
Great. All right. Well, thank you very much for coming on. And you can now retire. You can retire fully now. No more podcast.
Thank you, Glenn. Thank you, Glenn. I appreciate that.
Yeah.
It was fun. I enjoyed it.
Oh, well, thank you very much for coming on. See you later.
See you later. Bye.
Bye.